Welcome friends. Today we will discuss the C4 pathway. What is C4 pathway and why we will learn it? Why we will study it? The question arises. Look, in C4 pathway, it's something related to photosynthesis, photorespiration, everything. So we will have to look back to our photosynthesis past. Then we will go to photorespiration. Then we will realize why should we learn this. In photosynthesis, you have seen that there was RUBP, that is ribulose bisphosphate. We are forgetting about the grana portion because in the grana photosystem 1, 2, 3 and 4 and 2 are there. Okay, forget it. Simply we will learn the Kelvin cycle. In Kelvin cycle we have seen that whenever CO2 comes in, RUBP we have seen it is a 5 carbon compound and CO2 is having 1 carbon. Then with the help of one enzyme, that is RUBP carboxylase, RUBP carboxylase, we have seen that we have produced, the plant produced phosphoglyceric acid. It is a 3 carbon compound. This phosphoglyceric acid was the first stable compound because we have gone through this, that when RUBP and CO2 mixes, there was an intermediate product which is 6 carbon compound, but that is not stable. So we have seen that PGA is the first stable compound and carbon dioxide, that the carbon has been incorporated, that we have gone through. Now this PGA was the first stable compound in photosynthesis in Kelvin cycle. And then this PGA goes through several steps to produce the RUBP again, PGA to PGALD and others, and it will produce RUBP again. And this PGALD, I am writing down, PGA to PGALD. Then out of 12 PGALD, 10 PGALD will go this way and 2 PGALD will join to form glucose. Will join to form glucose. Now you see why there is a need to write this one. Here, everything is going on. This is known as Kelvin cycle. This we have seen, and it follows this way and that way, producing glucose. Again, in photorespiration, what we have seen, that in case of increase in temperature, the concentration of oxygen increases. So in absence of any carbon, this RUBP, ribulose bisphosphate, breaks up. Okay, ribulose bisphosphate is a 5 carbon compound, and this breaks up to 2, that is 1 3 carbon phosphoglyceric acid and 2 carbon phosphoglycolic acid. This is phosphoglyceric acid is CH2OP COOH. This was the formula phosphoglyceric acid. This is phosphoglyceric acid. Again, this was in photorespiration we have gone through. This is known as phosphoglycolic acid. So it has ribulose bisphosphate is a 1, 2, 3, 4, 5 carbon compound. So ribulose bisphosphate having 2 phosphate. It is bisphosphate. So we used to call it as ribulose 1,5 bisphosphate. Now it is breaking down here. It is breaking down here. So this phosphate went there and another phosphate was there. So the formula is CH2OP and COOH. So this is known as PGA and this is known as phosphoglycolic acid. Okay. This phosphoglycolic acid was immediately converted to, this is known as phosphoglycolic acid is immediately converted to glycolic acid. And this glycolic acid with the help of peroxisome with the formula known as transaminase, it is becoming glycine. You have gone through this my previous photorespiration. Please go through, feel free to go through there. There I have elaborated everything. Now this phosphoglycolic acid is converted to glycolic acid and glycolic acid is converted to glycine with the help of peroxisomes and the process is known as transaminase. Then glycine is immediately converted to serine with the evolution of CO2. With the evolution of CO2. The question is here. I told in my photorespiration class that plant is losing CO2 for this temperature increase and O2 concentration increases. So RUBP carboxylase acts as an RUBP oxygenase and it breaks up the RUBP into phosphoglyceric acid and phosphoglycolic acid, which is converted to glycolic acid, then glycine, and serine with the release of CO2. This is not negligible. In book, it is written that the expense of this evolution of CO2 is not trivial. It is not negligible. You should write. It is not negligible. Why? Because the hard and carbon by photosynthesis, the plant has incorporated carbon. They are losing half of their carbon. That is, biomass is decreasing. So plants are losing this carbon in the form of CO2. To overcome this, we will need C4 pathway. Now, in examination hall or in your examination year, you will be given with a paper, and almost all the students used to avoid this, and they used to attempt some other question, 1 or 2 or 3 or 4 or ABCD. But nobody starts with this because it becomes a little bit complicated. Why should I have to start, where should end, and this and that? It is very simple. First, you will write that in CO2, in normal photosynthesis in Kelvin cycle, this RUBP carboxylase, RUBP is produced, they produce PGA. Then PGL to PGLD, and it follows the Kelvin cycle on one hand, and other hand glucose is produced. Again, to write in the second paragraph, again, in photorespiration, what we have observed that the plant, in absence of carbon dioxide, the RUBT itself splits into two. One is 3 PGLD, phosphoglyceric acid, and another is phosphoglycolic acid, and this glycolic acid, glycine 7 to write that one. And with the evolution of CO2, plant is losing the half of their CO2, assimilated CO2 in the form of CO2 for photorespiration. And to overcome this, here starts the C4 pathway. C4 pathway is also known as beta carboxylation pathway, and it is also known as cooperative photosynthesis. We'll come later. Now, we'll start with C4 pathway. Many plants, usually in Gramineae, Cypressae, and in some monocotyledons, dicotyledons, both plants, many plants have evolved or adapted themselves to overcome these losses of CO2. Plants of sorghum, G-maize, and some other amaranthus plants in dicotyledons. So many plants, they have overcome these losses. How? These photorespiratory losses has been overcome by the plant itself. In 1966, this is known as Hatch-Slack pathway. Why? Because Mr. M.D. Hatch and C.R. Slack, these are the two scientists, while they were working on photosynthesis in sugarcane plant. Sugarcane is known as, scientific name is Saccharum officinale. Sugarcane is known as SACCHARUN. Saccharum, OFFICINALE. Saccharum officinarum is known as sugarcane plant. So these two people of Australia, they were experimenting something on photosynthesis on this Saccharum officinale plant, that is sugarcane plant. They have reported, what they have reported? They have reported that the first stable compound, it was PGA. Their PGA formula is, I have told you, PGA is CH2OP, 3 carbon compound. That is 3 carbon compound, and this is 3 carbon compound. This is known as PGA. These two persons, while experimenting on photosynthesis on Saccharum officinale or sugarcane, they have found that instead of 3 carbon stable compound, they have found 4 carbon compound. How? That is something different with that. What is the name of that 4 compound? Compound is known as oxaloacetic acid. Anyway, we will come later. 4 carbon compound is COOH, another carbon, another carbon, another COOH. This is the formula CH2. This is known as oxaloacetic acid. It is a 4-carbon compound known as oxaloacetic acid. So instead of this first stable compound in normal photosynthesis, they have found in Saccharum officinale another compound having 4 carbon, the first stable compound. First stable compound. Because this is the stable compound which they have experienced or they have tasted. So they said that it is a 4-carbon compound, so they named it C4 pathway. And this is a 1, 2, 3. These are known as C3 pathway. C3 plant, these are called C4 plant. The plant which produces first stable compound like oxaloacetic acid is known as C4 plants, and the others are known as C3 plants. Clear? According to them, according to these two persons, they have seen that primary carboxylation, that is, it is because of carboxylation, that is, input of carbon with the RUBP, RUBP carboxylase, the PGA is produced. They have reported that CO2 is coming and it is accepted by a 4-carbon compound, 3-carbon compound. Here it was 5-carbon compound, and here it is a 3-carbon compound known as phosphoenol pyruvic acid. What is phosphoenol pyruvic acid? We have to go back to glycolysis. You please go to my early chapters in glycolysis, wherein I have said that a formula known as G square, F square, and P6. I am just recapitulating. G square is glucose, then glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate. It splits into 2, PGALD and dihydroxy acetone phosphate. They are interconvertible triose phosphate isomerase. Then I have told you that P6, PGALD, then PGA, then 1,3-di-PGA, then 2-PGA, then phosphoenol pyruvic acid, then pyruvic acid. 1, 2, 3, 4, 5, 6. So there we have come across phosphoenol pyruvic acid before producing pyruvic acid. This phosphoenol pyruvic acid, you see, this is 3-carbon, and again all these 3-carbon. These are all 3-carbon compound. So instead of acceptor molecule RUBP is the acceptor of CO2. Here the acceptor in Saccharum officinale in sugarcane, that PP, phosphoenol pyruvic acid, is 3-carbon compound. So 3-carbon compound and 1-carbon compound. How much? It will be a 4-carbon compound. So phosphoenol pyruvic acid is producing a 4-carbon compound. 4-carbon compound. Clear up to this? Okay. Now we will go to further detail. This phosphoenol pyruvic acid In order to know this phosphorylated and this reaction and that reaction, we have to go through the leaves of this plant. We have to go to the anatomy, the leaves of these plants. How? Let's see. Take a leaf. Suppose this is a leaf. You take a leaf and you cut this leaf in this way, transverse section. So I am writing the leaf on that way. It is the upper portion and it is the lower portion. Not in this way. I have cut the plant in this way. Okay. So this is the upper portion, this is the lower. So it is known as upper epidermis. And this is known as lower epidermis. And the leaves, the plant takes water through the vessel known as xylem. So xylem is coming to the leaf. So xylems are there and phloems are also there. So these xylems and phloems are like rods. This is a rod. This is phloem rod. This is xylem rod. So these rods, if I cut it in this way, how it will look? It will be looked as round because these are the rods. Suppose this is my leaf. These are the rods, xylem, phloem. I am cutting here, so it will be round in shape. So let me write here. This is a round, round, round. Here also, round, round. These are all xylems for the purpose of our study. And there are phloems also. These are phloems. These are phloems. So xylems or phloems are there. And in C4 plant, these xylems and phloems are collectively called vascular bundle. These vascular bundle are specialized because they have a sheath around them. They have a sheath around them. These are all bundle sheath cells. These are all bundle sheath cells. Xylem, phloem, cambium, whatever it is. So these are the canal like this. Water is coming through the xylem and the produced or the products goes through the phloem. So in the leaf of this C4 plants, it has, and we have seen that here are some cells. These are all mesophyll cells, and in normal you will say there are longitudinal cells known as palisade parenchyma, and these are called spongy parenchyma. In C4 plants, these palisade cells or palisade tissue are absent. But it is filled up with spongy parenchyma or mesophyll cells. You can say these are all mesophyll cells. In C4 plant, the vascular bundle is covered all around by bundle sheath shells. Okay. So I am taking this out to give you a better idea. This is xylem phloem. Bundle sheath cells are like this. These are bundle sheath cells. And in between here we have seen xylem. Let it be 3 xylem vessels. And phloem, let it be 3 phloem. In the bundle sheath cells. And above the bundle sheath cells, there are mesophyll cells or spongy parenchyma, but spongy parenchyma containing the chloroplast are called mesophyll cells. These are mesophyll cells. So this is mesophyll cells containing chloroplast, and this bundle sheath cells also contains chloroplast. Now what happens? In the mesophyll cells, there are chloroplast. Chloroplast we have seen, we have written in this way chloroplast, something like that. There are grana. Remember, there are grana, there are stroma also. The basic difference is that in the bundle sheath cells, there are chloroplast, but here the chloroplast this is a double membrane. It has less developed grana, but it has maximum stroma. It is having less developed grana in bundle sheath cells. This is bundle sheath, SHEATH sheath cells. The bundle sheath cells have less developed grana. What grana does? Grana does the light reaction, is not it? And the stroma acts for the dark reaction or light independent reaction. On the other hand, these mesophyll cells have well developed grana. These grana are well developed. Grana with thylakoid. So these grana are well developed here. So light reaction may take place. Here dark reaction will prevail or will be prevalent. Now what happens? Whenever they have tested or they have reported that this anatomy of the C3 plant and C4 plant is known as Crange anatomy. What is known as Crange, Crange anatomy. This is known as Crange anatomy. They have seen that in C4 plant, these are prevalent. In C3 plant, the mesophyll cells contains this, and this bundle sheath cell also contains, but there is no difference in this grana. Now what happens? This phosphoenol pyruvic acid this is mesophyll cells. This is mesophyll cells. These mesophyll cells have chloroplast in it. These mesophyll cells These are the mesophyll cells. They here are the stomata. From the stomata, CO2 enters. CO2 enters through the stomata. And this is accepted by phosphoenol pyruvic acid, which we have gone through in the glycolysis. It is a 3-carbon compound. So here CO2 acceptor is paper, that is phosphoenol pyruvic acid, while there CO2 acceptor was RUBP, ribulose bisphosphate, a 5-carbon compound. Here it is a 3-carbon compound. So they have seen, or they have reported, that when CO2 enters through the stomata, it is accepted by PEPA, phosphoenol pyruvic acid, with the help of enzyme PEP carboxylase. PEP carboxylase acts here. And there RUBP carboxylase. Here PEP carboxylase acts on CO2, that is 3T and 1C, produces malic acid. Malic acid, it is a 4-carbon compound. Clear? So what these two persons, Hatch and Slack, have seen, that CO2 is accepted by PEPA. So PEPA is a CO2 acceptor, but it is not stable compound. Thus PEPA then produces malic acid. I'm very sorry. PEPA then produces oxaloacetic acid. Oxaloacetic acid, a 4-carbon compound, which you have shown here. C. This is the formula of oxaloacetic acid. COH, C double bond of CH2. C double bond of CH2. This is the oxaloacetic acid. So this oxaloacetic acid was first produced as a stable compound. So we should write that in C4 pathway or C4 plant, when CO2 enters, it is accepted by a acceptor, phosphoenol pyruvic acid, and with the help of this enzyme carboxylase, PEP carboxylase, OAA, the first stable compound is produced, that is 4-carbon compound, and hence the name C4 pathway. This oxaloacetic acid then converted to malic acid. Oxaloacetic acid then converted to malic acid. Malic acid we have gone through. We have seen during our course of respiration, we have seen that pyruvic acid, then acetyl coenzyme, it enters the mitochondria. In the mitochondria, there was, what is that? Oxaloacetic acid. Along with these 2 carbon, they produce a citric acid, a 6-carbon compound. And this OAA is produced from malic acid. Malic acid was produced from fumaric acid. Fumaric acid was produced from succinic acid, then succinyl coenzyme, then alpha ketoglutarate acid, then 6-carbon isocitric acid, then alpha ketoglutarate acid. In this way, Krebs cycle goes on. You go through Krebs cycle, you will find that oxaloacetic, prior to that malic acid. So here, oxaloacetic acid is converted to malic acid. It is also a 4-carbon compound. The malic acid this malic acid can enter into the bundle sheath cells, which have less developed grana and strongly thromboplastin is there. So Kelvin cycle goes there and it is full of Kelvin cycle enzymes. So let me write it a little bit bigger. These bundle sheath cells. This is the bundle sheath cells. Okay. This is the bundle sheath cells. These bundle sheath cells, through this bundle sheath cell aperture, malic acid enters. Malic acid enters into the bundle sheath cells which is around the vascular bundle. This bundle sheath cell is full of chloroplast with less developed grana. Now malic acid when enters, it gives out CO2. It produces pyruvic acid and gives out CO2. This is the main important point. This malic acid, when enters into the bundle sheath cells, it produces pyruvic acid and it gives out CO2. That is, CO2 is given here in this cell. I have told you that full of Kelvin cycle enzymes are there. This CO2 is accepted by RUBP. This RUBP and CO2 with the help of RUBP carboxylase, it produces PGA, PGA2, PGALD, PGALD, PGALD2, RUBP again and glucose again. What? Glucose, C6, H12, O6 is produced. So here Kelvin cycle goes on. So this carbon is accepted here because the C4 plant has made the inbuilt mechanism of overcoming the losses of CO2 and they circulate through the Kelvin cycle. On the other hand, this first pyruvic acid goes back to, this is known as mesophyll cells. They goes back to mesophyll cells. Then PGA is converted to phosphoenol pyruvic acid with the help of ATP. Here ATP is required. So for 6, 12 ATP is required. 12 more ATP is required. But in the earlier Kelvin cycle we have shown that when this PGA was produced, 12 ATP was required to convert PGA to 1,3-dipga. Again, 6 ATP was required from ribulose monophosphate to ribulose bisphosphate. The 12 plus 6, 18 ATP was required, 12 plus 6, 18 ATP was required for that C3 pathway. And here, out of this 18, another 12, that is 18 plus 12 is 30. So 30 ATP is required for this. But they are overcoming the loss with the help of this. Now, this pyruvic acid is going back to the mesophyll cells and reconverted to PEPA with the help of 12 ATP. On the other hand, CO2 is accepted by RUBP and the Kelvin cycle goes on producing glucose. Glucose is soluble in water. This water, glucose, goes through this. These are called pH, LOEM, phloem. These are xylem, water comes in. These are xylem, water comes to the leaf through the xylem and the products. Glucose is soluble in water, it goes through phloem to different storage organ of the plant and get there stored as in the form of starch by losing one molecule of water. So in this way, they have overcome these photorespiratory losses. So why we have read it? C4 pathway to see that how the plant has made the inbuilt mechanism of overcoming the photorespiratory losses, and hence the C4 pathway started. C4 pathway is known as beta carboxylation pathway. There are two carboxyl groups, and it is known as cooperative photosynthesis. They are cooperating the photosynthesis by doing this. So this is cooperative photosynthesis. That's why we have gone through this. Till then, what is the C4 pathway or HSK pathway is over. So what I have said, you take a pause and go through the sentence I have said. Write it down. You will see that there is a sequential step in which I have narrated this. So whenever it will come in the exam, you narrate the way I have told. Simply, and you will get the marks. Don't attempt the question which is attempted by so many students. They are avoiding, almost all students are avoiding this. You should attempt this. Thank you very much. Thank you.